There we go. Okay. So this is the enzymatic oligosynthesis project. First, we're going to talk about what exactly oligos are and what they could be used for. So oligos are short, single-stranded pieces of DNA. They're usually used for like primers in order to do site-directed evolution or to do amplification through PCR. You could also have certain single-stranded DNAs that fold in on itself and cause protein interactions. So they're usually known as ribozymes, riboswitches, and so on and so forth. You also, let's see, you also have the ability to, in this talk, to be able to elongate it and ligate a bunch of short pieces together in order to create entire genes that code for proteins.
Mhm.
So we're going to be talking about the basics of the traditional method of synthesizing oligos, which is known as the phosphoridomide method. So they're synthetic precursors to NTPs, the ATs, Gs, and Cs, and they're stuck together using a chemical method that uses harsh, flammable, and toxic chemicals in order to stick them together. So here's the highlight of the basic synthesis cycle. We'll delve more into that a little bit later. So we'll start at the beginning with the solid linkers and support. So in order to be able to elongate the the oligo, first we need to be able to salvage the pieces of DNA that we want to, while being able to wash away all of the unreacted bits. So the way that most commercial places use is they use CPG, which is a controlled pour rated glass. So it's a glass ball. It's got a bunch of tiny holes in it. The holes are very uniform, from anywhere from 500 to 3,000 angstroms. So 500 is usually used for shorter segments, like we're talking about 200 or less base pairs, whereas the 3,000 is usually used for, up to about 300 or so base pairs. You also have aminomethyl multiporated polystyrene, which is the exact same idea, except for instead of glass, they use polystyrene. So once you have the ball, the support of interest, you then use this 3-aminopropyl trithioxysilin to create an aminopropyl chain. So the aminopropyl chain will actually uh, click on to the 3' end, and kind of on the 3' end, and then hold everything in place, so that way you could do elongation from the 5' end. You also use anhydrous acetic acid, so super strong vinegar, in order to cap all of the amino groups on the the 2' end, where the amino acid is on each one of the NTPs. So after you have it supported, then you want to cap the 5' end in order to stop unwanted reactions. So as you can see right here, this is the example of the support linkers on the 3' end. And you have here on the 5' end this DMT group. which is a different DMT than some people are thinking. So this DMT group can be removed by using a light acidic condition. So that way you could continue to elongate the chain. And these are, of course, the examples of the nitrogenous bases that the anhydrous acetic acid is there to protect. So let's see. So here's the overview of the process. So we have right over here the linked 3' end of the NTP. We have the 5' end blocked. First step is to deprotect the 5' end. So they usually use trichloroacetic acid or dichloroacetic acid in order to remove the 5' end. So you have the exposed alcohol group. Once you have the exposed alcohol group, you go to the coupling phase. And side note, I talked to some pretty good chemists, and they said that the 5' end is way more reactive than the 3' end. So as long as you don't add any extra, like in excess of your reactant, it'll exclusively go to the 5' end most of the time. And the 3' is more reactive than the 2', especially if it's deoxygenated already. So you do the coupling, you add in another one just like this, but with whatever specific base that you want next. And then the coupling begins. It's usually done under acetonitrile, so anhydrous conditions, and then you use an acetic azole as a catalyst. And that will cause this bonding step right here, where you have this phosphate triester and a little scar right over there. So we don't want a phosphate triester. Obviously, we want like a regular phosphate group.
Mhm.
So we have to protect the amino groups, because if we went straight into oxidation, we could have side reactions and then destroy our chain. So we use a mix of acetic anhydride and methylimidazole or DMAP as the catalyst in order to protect all of the nitrogenous reactions. Then we go for an oxidation step. So use a weak base, pyridine, lutidine, or collidine in order to fully, let me move you guys over there so you can see, in order to add in that extra oxygen group. This scar just remains there, but once you go through amplification, it's it kind of gets itself gone. So I mean, it's not really that much of an issue. And the cycle repeats for your next base of interest. So now we're going to talk about TDT. So TDT, this lovely little protein right over here, is part of the X family polymerases. It has template-free activity and is mostly found in the thymus and lymph nodes. So basically, what TDT does is in a VDJ recombination for antibody production, you have you have the steps where you have the complex that comes in, cuts off two of the ends, and then you have this hairpin structure. Then you have the Artemis complex that comes in to remove the hairpins and create blunt ends. That gets activated by phosphorylation from the previous complex. And then TDT comes in over here and it's like, hey, there's a break. I got him, coach. So it just starts adding in random nucleotides, just whatever happens to be floating around, and just starts throwing them on. And then if enough of them match, they come together. And then if there's any extra unmatched nucleotides, then it just gets snipped off by this other protein complex. And then you have the variations, which gives rise to most of the activity for antibodies. Um, so TDT also has the ability to patch up single-stranded breaks. So if you have like a template and then you have two other pieces of DNA on either side, then TDT can come in and then insert the missing piece. So not too useful, but maybe there's a niche ligation application that it would be good for. So worth mentioning. So now we're going to talk about how TDT can be used for oligosynthesis. So there's two techniques right now in order to use TDT for oligosynthesis. We have the reversible terminator, which is my favorite, and we have the tethered approach. So the reversible terminator, it's non-dangerous synthesis. The most dangerous thing that I'm looking at is probably guanidinium chloride, which I mean is not dangerous, but you don't really want to like flush it down the toilet or anything like that. However, the DNTPs with the 3' protecting group, it's an amino oxy group, is pretty hard to get a hold of. I managed to track down a few companies, however, for each one, like if you want DATP, DGTP, whatever, it's in the neighborhood of about $400 to $600 for 5 moles of each. So
Oh,
uh,
yeah.
yeah, so I mean, you're talking about $2,000 to $3,000 just to get a hold of these, unless of course they're willing to sell you a smaller volume, or you can make it yourself. Plus, they're very selective on who they sell to. There's not like it's not in high demand right now. So, but the cycle goes. You have the oligo primer, and then you have the TDT that comes in. It's going to pick up one of these NTPs with the 3' blocking group, and it's going to attach it onto the 3' end. So 3' gets attached to 5', and then it drops off. But since there's the blocking group, it can't grab another and keep adding on the NTPs. So after you do that, all you have to do is get rid of the TDT, denature it through either heat, acidity, or washing step, and you do a deprotection step. With this amino oxy group that we're using for the 3', we can get it done with a little bit of heat around 50 degrees centigrade or Celsius, excuse me, and then a light acid wash. And that will open up the 3' end, and then the cycle starts over again. With the tethered DNTP approach, you're going to actually use a little linker. Some of the linkers was like PEG, but it was short PEGs. So short PEGs can be pretty toxic. So you could end up with some organ complications, but as long as you take precaution, it should be okay. The pros are the chemicals are easily accessible and it's cheaper this way. But in order to deprotonate with the short polyethylene glycol linkers, you have to use beta mercaptanol, which beta mercaptanol is also potentially fatal if you get on your skin. So you have to work with a lot more dangerous for human chemicals. So that's why I don't really appreciate the tethered DNTP approach. However, maybe somebody could figure out a way in order to, make it a little less dangerous. I did hear of one group that were using a different linker that was photocleavable. So you just like expose it to UV light, and then it'll release the NTP. However, that takes about 30 minutes in order to achieve that. But yeah, the basic extension reaction is you have the primer, you have the complex over here, then the TDT and the nucleotide gets added onto the primer, and then you do your washing step in order to get rid of all the rest of them, and then you cleave the linker, and then you start all over again. There are several companies that use this approach. Actually, the tethered DNTP is the most popular approach from the companies that I've researched, but I think the reversible terminator approach is better in basically every way. So I think that's just because this is easier and slightly cheaper, but more dangerous. Um, so now we're going to go through engineering issues. So we'll start from the left. TDT engineering, which is what we're going to be getting into in the next couple of weeks. So after we get TDT, the two biggest issues are thermostability. Definitely if you're using the 3' protected DNTPs, because that needs to be done in higher temperatures, and also as a side product, the higher the temperature that you can raise it, the less likely that you're going to be causing hairpins and other, folds that you don't want to. So you have the ability in order to print longer sequences than you would at lower temperatures. Also, TDT is not accepting or readily accepting of the 3' blockers. So some directed evolution has to be done in order to make sure that the TDT will accept the 3' blockers and to increase the thermostability. There's been several companies that have been able to achieve this feat, so I mean, definitely doable, not a pipe dream, but unfortunately, they all want quite a bit of money for that. So the only way that we'll be able to do that is if we do it in-house. Then we have the apparatus engineering. So the basic steps is we need to have DNA binding. We need to be able to wash away everything that we don't want, and then we have to have be able to deprotonate it and increase the thermal activity, so that way we could deprotonate everything and be able to print longer sequences. And then with nucleotide engineering. So this is if we want to try our hand at making our own 3' protected DNTPs, we need to figure out how to add the protecting group, obviously, or we could source the DNTPs with the 3' protecting groups, which I also mentioned. I've done quite a bit of work on both. So now we're going to talk about TDT engineering and its challenges. So over here, like I mentioned, oops, screen sharing is stopped. Okay, share again. Beautiful. Okay, so over on this side, like I mentioned before, at lower temperatures, there's a good chance that hairpins will be formed after a certain length. And that length is kind of, up to chance, depending on how juicy rich your sequence is. And, a lot of luck on top of it. But generally, you can't print more than about 200 to 300 nucleotides before you run into a hairpin that just breaks all activity. So that's why TDT usually doesn't work above temperatures of about 40 degrees Celsius because the body's at 37 degrees Celsius. So I mean, you have to be running a pretty good temperature in order to hit 40 degrees, potentially a lethal temperature. So TDT has absolutely no reason to, survive temperatures higher than that. So to avoid hairpin formation, we really want the reaction temperature to be above 60 degrees, about 60 to 70 degrees. That's, if you remember from PCR, you know, that's about the Goldilocks zone for elongation without having to worry about hairpins being formed or, like the strands and re-annealing or anything. So we definitely want to get TDT to, be able to survive somewhere in the neighborhood of 20 to 30 degrees more than it wants to. And there has been some researchers that have been able to get some pretty good activity in about the high 50s range. So it is potentially possible. Also, wild type TDT, like I mentioned before, doesn't accept the 3' block NTPs. So we have to do some work in order to make sure that we have a variant that is, a little bit more accepting of some weirdly shaped molecules. So, side note, TDT has 2 to 3 isoforms depending on which organism that you're talking about. In most organisms, mice especially, you just have the TDT long and the TDT short version. And in humans, you have TDT long 1, TDT short, and TDT long 2. So there's not too much difference between the TDT long and short. They do basically the same. Some people are speculating that the long version has proofreading abilities, as exonucleitic activity. Uh, so let's see. As far as doing the approach, we could do purely synthetic, possibly, which is contrasting consensus sequences, finding lesser conserved regions, and doing targeted mutations. Or we could do a directed evolution approach, grow in extremophiles, or at least like we could probably get away with just regular like E. coli, K12 turbo, something like that. There was actually a paper that I was referencing that did that, and then they just raised the temperature when they were doing the checks on the catalytic activity. But and then incur random mutations using you could either use a site-directed approach by by using primers in order to do that, or the paper that I was referencing that seemed like they had a pretty good amount of luck used leaky polymerases, error-prone polymerase in order to do that. And you lyse the cells, and then you're left with a bunch of random mutations on each copy of the plasmid, and then you inject those. You do the transformation, then you check to see how many colonies that you have that were successfully transformed, and you lyse each and every one of them and check to see if they have a useful variant. And then if they have a useful variant, then you keep that one, and the ones that don't work, you just toss out, and then you repeat the cycle until you have something that's good enough. Um, so that's the overview of that part. And then for the apparatus engineering, so I'm imagining we would have a thermocycler or a hot plate or something that can keep a consistent temperature, preferably with a little bit of cycling abilities, but maybe not necessary. And then because with the linking arms for the traditional phosphoridamide method, it's on the 3' end. So that would be difficult for us, considering that TDT likes to link 3' to 5' instead of 5' to 3'. So we can't use their sulfonyl linkers in order to be able to hold it onto the controlled porated glass. So a potential workaround that I was thinking of is that silica and the controlled porated glass has an affinity under guanidinium hydrochloride buffer to attract the DNA, with the longer chains, of course, being preferential to the shorter chains or the single NTPs. So they do have an electro or a magnetically doped controlled porated glass that we could potentially use. So my hacky workaround was we could do a guanidinium hydrochloride buffer that'll attract the bigger pieces of the oligos, and we'll be able to wash away the rest, the ones that we don't want, by using an excess of the guanidinium hydrochloride buffer. And then once we do that, we could take it out using like an electromagnet, and then we could wash the rest of the buffer and then do the deaminoxylation in order to get rid of the 3' protecting group. And then during that step, because the concentration of guanidinium chloride is going to be low, that will also elude all of the oligos that are attached to it. However, I have not tried that, and I suspect that that approach may not be a very good one. So, not married to this one. Some further research needs to be done. But the main idea of how the machine would work is you'd have to add the CPG and primer to a plate, and then and then you go to the automated part after that, where the machine adds the DNTP of interest, one from each one of the 4 tanks that you have, to the plates. Then you add the CPG attractant, which is the guanidinium hydrochloride buffer, and then the the oligos get attracted to the CPG. You lift the CPG out using an electromagnet from the reaction. You wash and drain with the guanidinium hydrochloride. You do a mag release, and then you amino-oxalate them. You remove the 3' protecting group, and that releases it from the controlled porated glass. And then you start again, and then you add your next NTP of interest. And then as far as nucleotide engineering, I would rather not. There's a handful of companies that produce them, but it is pretty expensive. So it would be nice to have some cheaper alternatives. So this is the first of the potential pathways I was looking into. So basically, you have your regular run-of-the-mill NTP over here. And first step is you want to get rid of this phosphate backbone. So you use phosphatases in order to be able to get rid of it. The actual phosphatase I'm planning on using is SAMH1, I believe. I have it written down somewhere. But that actually cleaves all 3 of the phosphate groups at once. Then you're left with the 5' unprotected. Then you need to add the DMT chloride in order to have this protecting group for the 5' end. And then the paper that I was referencing used what's known as a Mitsunobu inversion, and they did that twice. Added on this thing over here. I forgot how to even pronounce what that's called. And then you would end up with this intermediate, and then you'd get rid of the 5' protecting group and blah, blah, blah. But that is a complicated pathway, and most of it is actually patented. So, we could do that, but if somebody did that, they wouldn't be able to sell from that method unless they kept it really hush hush, So plus, it's not very efficient. These Mitsunobu inversions, you're talking about you're getting rid of 70% of your product each step. So I did some more digging, And and then, so this is a screenshot of the paper that I was referencing for the above synthesis, and they use these Mitsunobu inversions over here, and they use this explosive compound, or actually that's just methylamine, but they use methylamine, which just so happens to be a Schedule 1 precursor for methamphetamine production. So if you use that, you're going to make friends with the DEA, but that's fun.
Yeah.
But, oh yeah. So I did some more digging, and then I found this reference over here. So basically, instead of doing the Mitsunobu reactions, they decided to have it all under a brominated environment. So they used this explosive compound over here under a brominated environment in order to be able to add it to the 3' end. And they did no further reductions or anything. That's just a blocker. But the way that you get rid of that blocker is not very attractive. You have to use some pretty harsh chemicals in order to be able to do that. So plus it's a lot bigger than the blocker that I'm looking at. So you'd have to do a lot of work with TDT to even have a a prayer of being able to get it to accept this group. Um, so yeah, the pathway was modified over here, um, and I was able to save a couple of steps. Um, uh, or wait, that actually goes later. So I found out that this reference over here actually had a dye linker onto the added onto the base. So this extra protection steps over here is superfluous. It's not going to mess with anything. So this pathway was just getting rid of those superfluous protection steps, right? And then after we got to this, I consulted with a couple of chemists, and they said that if we switch from the first pathway to a brominated version, and we keep the TBAB as the environment, we could probably switch on this intermediate from the first synthesis pathway, and then be able to do the methylamine reduction, the methylamine and the I can't think of what that's called off the top of my back, reduction, in order to get to the product that we actually want to, this amino oxy group right there. So in addition, because these numbers down here show the known yields. So from the second paper that I was referencing, they say that they have a 95% conversion between this, the unprotected 3', to this intermediate group. So as you can see, where we're really lagging is this step, the Ludwig-Exton triphosphate synthesis. So to further improve this pathway, I'm thinking about trying to use kinases in order to add on the phosphate groups, since we're using phosphatases to take them off in the first place, and we might be able to get a much higher yield. So that's the next low-hanging fruit that I could see. And that that's all that I was thinking about so far. Does anybody have any questions about this?
Where did you get a chance to test this out in the lab?
Uh, this synthesis pathway? No,
Yeah.
I have not. Yeah, it requires a bit of HPLC work. I can definitely test up to about here, and then I could send it out for NMR,
Okay.
because that's basically the only thing that we could do. However, the rest of the chemicals that we need outside of methylamine, I mean, methylamine's pretty easy to get, but we're going to be making friends and stuff. Um, it's none of these are really toxic. The biggest thing that I'm worried about is TBAB, but TBAB isn't even that bad. But yeah, I was crunching the numbers and it looks like I'm going to need somewhere in the neighborhood of about 3 grand or so in order to check to make sure this pathway works in between the resins and columns that I'm going to need, as well as some of these niche chemicals. So we are doing some crowdfunding campaigns with experiment.com. And I have some friends that said that I could use their HPLC. So I would be highly interested in verifying this pathway and then figuring out exactly which is the best ways of purification. Because for most of these steps, I have more than one potential way to do purification. So after we have that, we verify that it works and we know what's the best way in order to have the best yields possible. Then I was going to have this open source and be like, anyone that wants to make it, please make it, you know?
I know you'll make friends with the methylamine, but how much would you need?
For methylamine, not too much actually. Like I don't really need an excess. Man, it kind of depends like how much I'm doing at a time. But if I was using like uh yeah, if I was using like a mole at a time, then I would probably need like, you know, right about a mole, maybe a mole 0.2 or something like that to have just a slight excess. So I'm not sure how much methylamine weighs, but yeah, it's chances are it would be like 10 grams would be in excess. That would give me some time to like, you know, like fuck up a couple of times.
Well, that's what experiment's all about. Yes. Okay. All right. All right. All
Let's see. Anything else that any other questions that we got for right now? All right. Well, I'll take that as we're good for now. So I am going to stop sharing and recording, and then we can talk about some of the updates. Let's see. Where's the recording button?